Welcome to NPTEL certification course on Pharmacognosy and Metabolic Engineering. So, now we will go to lecture number 38, where I will talk about the biosynthesis of monoterpenoids. So, in the lecture number 37, I have drawn a range of cyclic as well as acyclic monoterpenoids. That means, to show you the product diversity, and most of which are components of flavor or fragrance. So, now we are starting with again the product diversification but here monoterpene synthase will come into the picture. That means, starting from the substrate GPP, how monoterpene synthase contributes to single product or multiple product formation depending on the species. So, that is our first part of discussion, and the second part is that I will talk about the general reaction mechanism catalyzed by monoterpene synthase. Let us go to the board. So, reaction products of two terpene synthases, if I write in this way, reaction products of two terpene syntheses. So, so I will start with this substrate, that is GPP. These two joins together and form these two isoprenes. So, these two isoprenes joins together and form the substrate for monoterpene synthase, which is GPP. So, this is now the structure of GPP I am drawing. So, GPP this is geranyl pyrophosphate. So, this will be converted to 3S linalyl diphosphate. So, this is common. So, all terpene synthase will, all monoterpene synthase will convert this. So, I put this as intermediate. So, this is 3 linalyl diphosphate. So, this will be converted into linalol. So, the product linalol is here. Although I have drawn this structure in the last class, but again I am drawing for your easy understanding. So, this is the problem with the boards. here will be one wedge here. Okay. So, this is the structure of linalol, LINALLL. The enzyme which catalyzes this reaction is linalol synthase, LIS. LIS stands for linalol synthase, and this was reported in Nicotiana alata. So, that means this is basically a single product catalyzed by linalol synthase. So, single product, and that was found in Nicotiana alata, particularly Nicotiana alata flowers. The floral volatiles is dominated by linalol, and that is catalyzed by linalol synthase. Now, let us see that there is another species which is called Nicotiana suaviolens. So, that species I write it in this side. So, in Nicotiana suaviolens, what happens that these linalyl 3 diphosphate undergoes or taking part in the formation of multiple products. So, multiple products means that I put an arrow here. So, put an arrow here. So, this is the enzyme is called CIN. CIN stands for cineol synthase. Cineol synthase. Okay. And the first product is intermediate which is this is called alpha terpinyl cation, alpha terpinyl cation. Sorry, alpha terpinyl cation. So, alpha terpinyl cation will subsequently converted into different products like limonene, like terpineol, and then it forms cineol. So, I will not draw all the structure because it will take more time. So, simply I will draw the major one. So, this is limonene, and this is alpha terpineol, and then this forms the cineol, which is the major one, 1,8 cineol. So, 1,8 cineole. So, the structure of 1,8 cineol I have already drawn yesterday, but in the last class, but I will like to draw it once again. So, this is basically the 1,8 cineol. If you put the eraser here, we can draw it like it in a better way. So, this is 1,8. Now, apart from these, there are other products which are also formed. For example, this also contributes towards the formation of mycin as well as beta ocimine. So, mycin and beta ocimine. And in addition to this also contributes to the formation of pinene. So, like alpha pinene and beta pinene. And finally, it also formed sabine. So, the yellow one is basically showing the monoterpene synthase catalyzed different reaction products, and that is localized in this species, that is Nicotiana suaveolens. So, Nicotiana suaveolens is the species where monoterpene synthase is operating. Nicotiana suaveolens. So, where monoterpene synthase is operating, and this is basically termed as syn because its major product was cineol. Now, let me tell you what are the percentage of different product formation. For example, it produces 60% cineol, only 2% terpineol, 10% sabinine, and 2% beta pinene, 2% alpha pinene, 10% limonene, 2% ocimine, and 10% mycin. That means, in Nicotiana suaveolens, the monoterpene synthase contributes to multiple product formation. So, I just put it here with this color, that is multiple product formation. And that is, and the main enzyme is cineol synthase, because it catalyzed it basically it led into the maximum formation of cineol, that is why it was named as syn, but it has multi product formation capability. So, that is the beauty of terpene synthase. So, one terpene synthase, monoterpene synthase, which is LIS, that only makes linalool, whereas another one CIN, it makes so many products. Now, both are present in Nicotiana, whereas in Nicotiana alata LIS is restricted, whereas in Nicotiana suaveolens this is restricted. Okay. So, once this is clear, so now we will go to the other aspects, okay, the reaction mechanism. Okay. Before I go to the reaction mechanism, I will also try to give you a brief idea about the different skeletal structures formed from this. So, that means, different monoterpene skeletons which form from this geranyl pyrophosphate by the action of monoterpene synthase. So, what I write here is the substrate is GPP and GPP makes that alpha terpenyl cation, and alpha terpenyl cation leads to formation of different products and different skeleton structures. For example, I will not draw all the skeleton structures because otherwise it will take time, but I can give references later. So, one is terpenen 3-wild cation, and the example of this product is beta phelendrin, PHELLANDRENE, beta phelendrin. The other example is terpenen 4-wild cation, and this leading to the formation of thuja skeleton, THUJA thuja skeleton. Okay, the structure of thuja skeleton is like this. Now, alpha terpenyl cation also contributes to the formation of boron skeleton. Boron skeleton. The structure is like this, and this also contributes to pinene skeleton. PINANE. This is pinene skeleton. From pinene skeleton, another skeletal structure formed, which is called Fenchene skeleton, FENCHANE, Fenchene skeleton, and okay, from pinene boron skeleton can also be formed. Okay. So, this is in general the different monoterpene skeletons formed from alpha terpenyl cation. Okay. Now, I go to I will give you some examples. Okay. So, let us go to the next slide. So, I have mentioned several monoterpenoids. Let me see how many slides I have. Okay. So, monoterpenoids. So, and different names I have told in the last class. Also, I have drawn the structure. So, here I am not going to draw the structure, simply I am going to write the names and the occurrence of these monoterpenoids in which plant, that I will mention. So, one is the geraniol, then comes to linalol, then comes to nerol, then first I am writing the name of the compounds, then geraniol or it is also called Z citral, then citronellal, then citronellol, then neral, or it is also called E-citral, and then say beta myrcene. You can also write thymol, and carvacrol. I have mentioned about carbone in the last class, and of course, the menthol. So, with this let me write the name of the plant. So, when I talk about linalool, I have also to write the linalyl acetate. So, linalyl acetate, okay, I will put the nerol here then, and the LINA linalyl acetate. Now, name of the plants, I put it. For example, geraniol finds in geranium oil, geranium species. You will also find in rose. Linalool you will find in lavender, you will find in jasmine, even you will find in tea, you will also find it in coriander. Nerol you will find it in rose oil. Linalyl acetate you will find it in lavender and bergamot. bergamot. Citronellol you find in rose oil, also you find in the oil of scented geranium. Neral or citral you will find it in lemongrass, and beta myrcene you will find it in hops and also in oranges. Menthol you find in peppermint, carbone you find in. So, this is one way we are revising the content of the earlier class. Spearmint, carvacrol you will find it in Indian prime rose, or it is called Plectranthus. Or it is also called as Indian prime rose. Thymol you find in thymus or thyme, scientific name is thyme. Citronella you find it citronella oil. geranium and citral you will also find it in lemongrass. Okay. This is in brief the different monoterpenoids and the plant species where they are occurred. So, once you know this, then we will go to the last part of the class, which is basically the proposed mechanism of this terpene synthase catalyzed reaction. So, in other words, the proposed mechanism of monoterpene synthase catalyzed reactions. So, the structure is this GPP this will form, later I will join with arrow. So, the first step is number 1, that is hydrolysis of diphosphate moiety, and this leads to the formation of a structure which is called resonance stabilized carbocation. Resonance stabilized carbocation, CARBOCATION. Okay. The second step is basically number 2, isomerization. So, this leads to the formation of so, this is intermediate capable of cyclization. So, this is formation of intermediate capable of cyclization. So, the next step number 3 is basically rotation around a single bond. Then in the next step, this is number 4, second diphosphate hydrolysis. Second diphosphate hydrolysis. Okay. That leads to formation of this structure. So, this is called again called carbocation. Carbocation. In the next step, that is step number 5, which is cyclization. So, cyclization next step hydride shift, step number 6. Number 7, second cyclization. And next this can lead to the formation of two different products. One may be number 8, which is deprotonation, and number 9, which is basically capture of the cation by water. So, deprotonation leads to the formation of this structure, which is this one, and next one is so this is called sabinin, and this is called sabinin hydrate. So, this is basically the proposed mechanism for the cyclization of geranyl pyrophosphate or geranyl diphosphate to sabinin and sabinin hydrate under the catalysis of a monoterpene synthase. That is, the monoterpene synthase enzyme catalyze these steps leading to the formation of sabinin or sabinin hydrate. So, it involves 8 or 9 steps. So, this is the beauty of the monoterpene synthase, and of course, complex, and those who worked with monoterpene synthase, they have elucidated lot of things which is also beyond of our understanding. Unless you do your research in this area, you cannot really understand that details. So, I try to put it the things in a very simplified way. So, what you need to know basically, now I will make it clear that you should know that there is a isomerization and there are cyclization. So, these two things are important: isomerization, rotation, cyclization, and you should also know this terminology which is called carbocation. Okay. So, this is in brief the mechanism of a monoterpene synthase catalyzed reaction, and in a previous slide I have also shown this structure, this sabinin here, sabinin, it is where I am showing 10%. So, this is how it is formed. So, this is just one example. Now, the limonene formation also follows a similar step, and then towards the end there is slight changes in the conformation, but obviously we cannot study all. So, this is one of the classic mechanism reported in a good review written by a subjects expert. From there only I have picked up and try to put it here for you for the class. So, with this I should end the class now, and let me see anything is left out or not. So, like what I have covered, let us go back. So, this is the reaction mechanism, this is the different monoterpenoids formed, and that is the diversity of monoterpenoids and the plant species it produces. Then different monoterpene skeletons formation, this is just for your information, and then in most important part is that monoterpene synthase can catalyze the single product or it can catalyze multiple products. So, here I have given you example of Nicosia and Alata, where single product formation by a monoterpene synthase, specifically called linalool synthase, and the other one is another monoterpene synthase which occurred in Nicotiana suaveolens, and that catalyzed 1, 2, 3, 4, 5, 6, 7, 8 products. And so that was named as cineol synthase, because cineol was the main product. So that is the beauty of monoterpene synthase. So product diversification originated from monoterpene synthases I have covered, and the reaction mechanism catalyzed by monoterpene synthase also I have covered. So with this I end this class. Thank you. Before I go off, so we will talk in one of the subsequent classes about some metabolic engineering application of monoterpene synthase in plants, particularly with the flowers, and also we will see that as I said before, that floral scent constitutes a major portion of monoterpenes. So, we will also study the physiology of floral scent formation, what happens, how flower emits scent, what are the components. So, that will also be interesting for you to learn, and it also has lot of commercial implications. So, in one of the subsequent classes I will cover that. With this I end this class. Thank you.